my name is Andrew Dufresne. I'm a senior tutor with Prep Matters. Typically, I do a lot of teaching with students one-on-one, but I also have been in the front of the classroom in colleges and at the National Institutes of Health, something a little more close to home given where I'm located. Today I want to just basically start off our AP Biology review in the time of the pandemic. It's a different test this year. Only the first 75% of content from your course, your AP Bio course, is actually going to be on the exam. More information is coming out from College Board. Definitely go to collegeboard.org to get updates on that. And if you want it trimmed down so you don't have to go through all of their text, just pop on over to prepmatters.com. We have a COVID response page to basically give you information on that. But some of the details we do know are that the test is going to be 45 minutes long instead of 3 hours. So that's not necessarily a bad change. But it's only going to be free response, no multiple choice. I'm less of a fan of that change. And we're not really sure how it's going to work out in terms of long and short response, free response questions and points and all that. Without further ado, let's begin. Today we're going to talk about the chemistry of life. You'll see here, just as I do on the course page, I list what it is we're going to be covering. And it's very different from probably the order that you've gone over this in class. You may not remember much of Unit 1 from the beginning of the year, but it's very foundational and it introduces a lot of terms and a lot of concepts that are going to be applied throughout the rest of the course and the content. And so I figured it's still important to start there. But then I thought it would be an interesting story to kind of just build up from there, from that very, very small, and then go through the molecular biology of genetics, and then the idea of heredity and inheritance from one generation to another, and then how that's all packaged inside of the cell, and then how the cell operates. The other units that we won't get to, at least at the moment, in this series of courses, the classes, are about natural selection and ecosystems, where they really get bigger. It's unfortunate that those units won't be on there because they really do help tie things together. But they essentially just continue to draw out that microscope, where right now we're going to start off at the really tiny atomic level, and then we're going to just bring it back until we're basically at that cellular level. And in your course, were it to even catch College Board's videos about any of this, when they complete the material, it gets up to the organismal level, populations, ecosystems, biomes for the entire planet. So today is going to be the chemistry of life. Okay, so what are we actually going to be doing? We're only going to be reviewing content. The expectation is that you've had this course, or that you are at least preparing independently for the AP exam as well. There's just simply no way I can take 75% of a full year course down into 6 45-minute segments. That's just not going to happen. So we're going to hit just some of the most important and far-reaching information. And then really we're going to move into these ideas of what does carry on from one unit to the other. What is core and fundamental to the AP Bio exam, and what do I need to keep in mind in order to really make sense of all this information and then apply it on actual questions. So that brings us to the idea that we're also going to talk at the end of each class about at least one free response question and FRQ, and talk about the strategies that you want to keep in mind when you approach these questions. So, as I said, highlights of the material, that's what we're going to be focused on. If you have any questions, if you have any confusion or anything like that, you see at the bottom of these slides, send us an email, classes@prepmatters.com. I love questions and would be happy to answer those. And I will try to make them trimmed down so that they get right to the heart of what it is you need to know for the test. But if you do have questions that are beyond the scope, I'm probably going to field those as well, just because I get so excited about talking about this stuff. So I also said that we're going to break down the exam into its core parts. We're also going to talk about the content and the trends that it presents. and how those connect across the different units. And then, as I said, we're going to work through those problems together. So with the recording aspect, you can pause it if you were to come back and review the questions afterwards, try to work through them, and then see, oh, that's what the scoring guideline says, and that's how I should work through that question. Okay, how are we going to actually go about this? What can I expect, in these classes and in these talks? Well, we have to speak the language. It's going to be a lot of vocab. There's going to be a lot of, what does this term mean? You want to see a word, a key term, and have that kind of be your trigger, and then respond with, okay, here's what I need to keep in mind when I'm dealing with that concept or going through that material. The same can be said for the action verbs or task verbs, I think as College Board calls them, and the free response questions. Identify versus explain versus describe. These all mean something in particular, and so we're going to talk about how we actually work through those as well. The manner in which these units connect, AP Bio, just like every other AP course in the College Board, they have these big ideas. Those big ideas are evolution, energetics, systems interactions, and the information and transmission of the storage and transmission of information. So that doesn't tell you too much by just giving the broad strokes, but as they come up, I'll describe those more. Otherwise, you wouldn't know what to hold on to right now for information. And the skills, we're going to talk a little bit more about those as well. It basically boils down to scientific method and skills that you would employ as a scientist, because that's the point of this class, is to kind of give you a foray into that world. And then, like I said, we're also going to go through those free response questions. I will say this, AP Central is something that College Board provides. Go to collegeboard.org and go to AP Central, and you will find a bunch. They've actually opened up a number of additional questions simply because of the situation that we have going on here with the pandemic and the change to school. But if you go there, you will find the questions that I'm going to be using, as well as a number of others. We're not going to be able to go through all of them. This unit's only going to have one at the end because it's much more foundational in terms of material, and there aren't a lot of that just center on this, but they apply these concepts. So here are the big ideas that I already had mentioned and some of the skills. So those skills, just to give you a little more idea of what I mean here by some of my notes, it's about making observations, conceptual understanding, visual representations, graphs, hypothesizing. What do you think would happen given what you have observed? Planning, how would you put together an experiment? What would be your methods? What are your things that you need to make sure that you have? Controls, independent and dependent variables. These are all terms that you will see again and again, and when you do, and they're applicable to a question, I will describe those more, but otherwise, that's what we're really talking about when we talk about plan. And then describe how do we basically take the data that we get from those methods and put them together on a graph accurately and well, and labeling axes, seeing general trends and all that, and then analyzing that. That both means what do we get out of the data, but then also what does that help us formulate for an argument in terms of the biological concept, in terms of what our prediction is or what our hypothesis stands in the end. If you do have any questions in the middle of the talk, there's also the Q&A feature. And when you send those, I see them, no one else does. And then if they're, like I said, if they're out of scope, then I may get back to them afterwards. But if there's something in real time, I'll just peek over to my question and answer window and then see if there's something else I have to explain on the slide more. So we will discuss in our, one question just came in was about what are some common mistakes that students make with some of these task verbs or something like that on free response questions, and how can we avoid those or write stronger answers. I'll get into that when we do the practice question at the very end, so you have something to hold on to. But essentially, it's a standardized test, so we can fall back on standardized methods and get some standardized strong results. What we want to pay attention to here is this oxygen atom in the center, and the hydrogen's here. It's bent, so there's an endedness. This is one end, this is another. What do I mean by that, and why does that matter? We're going to talk about some of the atomic structure of these, but essentially, we have these attractions that occur between water molecules. And this is something that is going to come up again and again as well. This little symbol right here means partially negative, and this one here means partially positive. That's because the electrons, we'll talk more about those in a moment, in the from the oxygen is attracting those more so, and so that makes this generally negative, and then the hydrogens from which it is pulling those electrons generally more positive. And as I'm sure you know, opposites attract, and so there's an attraction between these molecules. It's not technically a bond, it's an intermolecular force, but this would be one of the ways in which the molecule is polar and has one end here and then another end here, just as this is the positive and this is the negative for that attraction. This provides all kinds of properties to water, namely that it's cohesive, it likes to stick together. It's adhesive, it likes to stick to other things. It has something, because of this, this general interaction between the molecules and the strength of these bonds, it has something called a high specific heat capacity. So as it warms up, it doesn't quickly change temperature. That's a good thing because our body temp is one temperature, but we wouldn't want all of the water necessarily. The energy is there, but we wouldn't want to be walking around and have the water, you know, like for example, in the environment, all of a sudden change as quickly as something else, like I say a piece of metal. You heat that up and it heats up very quickly, its temperature goes up and it can burn things. Water is able to take that energy and just kind of move around a little bit more without necessarily changing temperature yet. And then also, because of these weird hydrogen bonds, solids and liquids for water are different from other elements. So in this case, when water freezes, it expands. So it gets less dense and then ice can float. So here are some of those key terms. You're going to see these slides every now and then. I am not going to be reading off of these. These are basically my way of just helping you out. All that stuff you just said, what's some of the most important stuff? The polarity or directionality or endedness of the water molecule, its cohesive and adhesive and heat capacity properties, and then something that it does in actual process in terms of state changes. This is important. You could think of even just, it's a good thing that ice does float. Otherwise, if it sank, all of the oceans would freeze and everything would die. And then we would have a very different whole situation. One really great question that I just got from a student was about when water goes up a tree, that we call that capillary action because it's moving up through these very thin little tubules and vessels. Is that cohesive or adhesive? That is cohesive, because what's actually happened, this also happens in transpiration, which is water leaving a leaf. And as that first water molecule leaves the leaf, it's literally pulling on the next one to replace it. And then that one is pulling on the previous one, and this is how water actually defies gravity and comes up. The tree or the plant is not going to catch as much water as the ground at its base. And so as that water goes into the soil and then gets taken up by the roots, it's able to feed the rest of the plant as well, internally and externally, because it is through cohesive flow, capillary action, getting pulled up through the vessels. Adhesive would be when water is sticking to something else. So if you've ever had a water ring on the bottom of a glass and on a table, and it's kind of hard to lift off of the table, that would be the adhesive property. So that was the importance of water, just the beginning of it. You can see it come up again and again. That's why I wanted to start there. I mentioned some terms about elements. So I want to talk a little bit more. I want to just backtrack a little bit and go into atomic structure, just to the point that you actually need to know for the AP Bio exam. So we've already talked about hydrogen up there in the top left over here. And we've talked about oxygen over here. We do not need to know about all these elements, but there are some key ones you're going to see happen and come up again and again. That includes carbon and nitrogen, phosphorus, sulfur, and chlorine, and then some metals over here, sodium, magnesium, potassium, and calcium. So these, sodium and chloride, that's table salt. Let's see, potassium, that's one of those great minerals that's in bananas, that's healthy. Calcium, same kind of thing, along with dairy products. So there's some biological relevance to a lot of these molecules. And we're going to talk about those as we go along, but I wanted to introduce them now so you had your bearings. Okay, so I mentioned before something about electrons. I didn't talk about anything else inside the atom. I want to kind of move through this a little faster. But essentially, as an example, here's an oxygen atom. This is the nucleus, the center, and it is made up of neutrons, the yellow ones, and protons, the red ones. That is not their actual color. This is just an artist's rendering. In fact, we call this the Bohr model of the atom, which you might remember from chemistry. This is also not actually how the electrons operate. Those are more of a cloud orientation, but for all intents and purposes, this is going to suffice for how you want to think about it. What else do we need to know about this? Essentially that the protons and neutrons are in the center and the electrons hover around the outside. And then, and that's why they're more prone for interactions and the attraction and repulsion between elements. And then also how you can have things like isotopes, which are the same element, but a different number of neutrons. Something that you may have come across, carbon-14, when we do carbon dating, that's what we're using. Radioactive water can sometimes have something called deuterium or tritium, and that's radioactive hydrogen. You also have, we determined some of the stuff about DNA using isotopes of nitrogen. So that's when there are a different number of neutrons. But the number of protons has to stay the same, otherwise it becomes a new element. Okay, so this kind of brings us to one of the first concepts that's going to happen again and again, particularly in today's class. This idea of smaller things come together to make bigger things. We have the electrons and neutrons and protons that come together to make the atom, and then we can have different elements come together to make compounds. As an example, this would be how water is formed. This would probably start off in the gaseous phase. You'd have a hydrogen gas, oxygen gas, and then that coming together into water. And all we're really wanting to get out of this is the idea that chemical reaction forms that brings these together, and then the nature of those different chemical reactions. In this case, and we're going to have that vocab slide in a moment, these are covalent bonds, not ionic. So the kinds of bonds, it's a matter of being able to give some depth to your understanding of the topics, but really what you want to focus on here is how smaller units come together to make larger units. Okay, so some of the terms again. Be aware of elements, atoms, and their different components. And then also here I am going to talk a little bit more about these bonds because I didn't mention those on the compound slide. I mentioned that in water, they're not ionic bonds, they're covalent bonds. That means that they are sharing the electrons. There's not a transfer of those electrons or a donation and acceptance. Something that does do that, table salt, which I mentioned before, sodium and chloride. Sodium gives up its electron and chloride takes it. This, again, will matter when we start seeing these ions in later units flowing in and out of cells and triggering various actions and processes. There are two kinds of covalent bonds as well because it is sharing, and there are different ways you can share, either equally or unequally. When something is shared equally, it's a non-polar covalent bond. Remember when we talked about polar endedness, this idea of one thing being a little more negative or positive than the other. And a non-polar covalent bond, not the case. Polar covalent, that's the example would be water there. There's a number of molecules that offer that, and that's important because the ever-presence of water in so many situations, including inside of our cells and outside of them, we want to know what's going to dissolve and break apart and what's not. Polar things, polar solvents like water, dissolve polar solutes or other particles. Non-polar dissolves non-polar. So something like oil and water don't mix. Water is polar, oil is non-polar. They don't really mix very well. Okay. So continuing on with that theme of building smaller units and putting them together into larger units, we have biological macromolecules. All that stuff about water is great. It is just the first player in this large story, And the elements are great, but they do so much more and achieve so much more and are so much more responsible for everything going on in the realm of biological life when they get together as these. So you can see here, these are actually carbohydrates, all of these on the page. These are actually, I'm gonna go ahead and pose a question and then see how quickly the Q&A can work in terms of typing the answer. And depending on how well that works, I may just go forward in the future and pause or set up some polling questions or something like that. But can anyone identify what these are, or at least even name the kind of carbohydrate that they are? Yes. So a couple of good answers. One in particular from Maddie, glucose. Glucose would be this one on this side. And then Jasmine also brings up another aspect, or Yasmin, apologies if it's pronounced that way, monosaccharides. So the mono one. Saccharide is another word for sugar. So these are monosaccharides. You do not need to know the structures for the test. That's the other thing I'm always going to do in my classes. I'm going to tell you when something, you don't need to know that. That's too much. I know a lot of students will feel like they need to, or that their class taught them such. It's not on the test. You can go to, again, the course descriptions from College Board directly. It's always nice getting information directly from them and see, in fact, that they have exclusion statements for the course descriptions, and it says knowing the structures of these sugars is beyond the scope of the test. So you're good there. These also can come together, like we said, little things come together, making bigger things that then have additional activity and important processes. They can form dimers, di for two. We're not going to get into the difference between why we're using di and not bi, as in bicycle, for two. It's a whole Latin Greek thing. But here, what we have are two glucose molecules come together to form this. Any idea on the name of this one? Yes. So let's see, Nolan. Yes, one of the dimers is sucrose, and it is this one here. Sorry, actually, let me rephrase that. This is maltose. This is two glucose molecules coming together to form maltose. The other two coming together are forming sucrose. So, okay. This up here, does anyone know the name of this one? This is fructose. That's the sugar that we commonly find in fruit. Yeah, they can basically come together independent of whether or not they're the same thing, and these form all of the different polysaccharides. There's mono, disaccharides, and then poly just for many. There are tri and other ones, but nonetheless, this is how those come together. Now, you'll also notice, and we're going to talk about this again on the next slide, but when these come together, if you imagine two of these next to each other, there's only one oxygen here in the middle, so it's missing one from the other side of the other molecule, and there are no hydrogens. There actually would be a hydrogen off of each of these little, we call them the vertices here. These are all carbons, and oftentimes we don't write additional hydrogens as we do up here, but that means that since there would be 1, 2, 3, 4, but there are only two unwritten ones, two hydrogens are missing, and an oxygen are missing. What do two hydrogens and an oxygen make? Water. So we took water out in order to make that bond. This bond is called a glycosidic linkage. Again, this is just to kind of give a little more flavor to how it is you're going to describe these things. But that's more of a, you can review that later on and stick to the big points here. Another large macromolecule that is very important, those are the carbohydrates. These are lipids or fats. So sugars and now fats. This right here would be an example of a saturated fatty acid. Oh, pardon me. And this right here would be an unsaturated fatty acid. Why? This little guy. This double bond right here basically means these carbons could have had more hydrogens. They don't, so they're unsaturated. That, as a chemistry fact, is great and all, but the real biological relevance is the fact that it does cause it to bend like that. And that's going to play a role later. So I'm trying to, again, point these things out now and then put them together, build just as the biological molecules do. I want to build these ideas together to get you to recognize now and then have to explain less of those details later. These fatty acids can come together to form triglycerides. Those are also some fats and foods by having this molecule glycerol attached to the end here. A couple of the things I want to mention about these, this hydrocarbon chain here is very different than this carboxylic group here. You don't need to know all the names of the groups, but this is why it's called an acid, because if you recall from chemistry, acids donate hydrogens. This hydrogen can come off because the oxygen's happy to take the electron and kick it off. This hydrocarbon chain does not have any very electronegative or electron-loving elements, and it's all non-polar covalent linkages, so that is non-polar and this is polar, and that is a huge component of this molecule. And then in glycerol, we still have a polar area here and then a non-polar end over here, and then you can also see, oh well, I only have one oxygen, and so there's an oxygen hydrogen here, and then there was another hydrogen over here. Oh, so each of these must have lost water. So you can see here, we call this dehydration synthesis. This is a reaction you're going to want to know because of how often it happens. You'll see it another few times even in today's talk. Okay, so again, you can come back to this. I want to continue on with our pace. A couple other lipids, phospholipids and steroids. Phospholipids, how do they differ from the previous triglycerides I mentioned? Well, they're not tri, but they have this additional phosphate group. There's that phosphorus that I mentioned at the very beginning in terms of elements you want to be aware of. And you notice that those also in that periodic table, they were all kind of grouped together. When you're grouped together on a periodic table, you have very common properties. And so this phosphorus, is the role that it plays, it's going to continue to play that role again and again. In this case, oxygen can form this complex around it, PO4. Well, it would typically be PO4 3-, but we have a couple other things bonded here. So this would actually be PO4 2- because of this one and this one. Either way, that is another polar group. You can see the charge even written on there on one end, and then the non-polar group over here. And then we have steroids. These are very different. You may recall something like steroids for something like weightlifting or something like that. This is something a little different than that. You can see these structures are very different. And I see some other questions coming in here, and I'm going to address those in a moment, so thank you for those. But cholesterol is a very important molecule. We often think we don't want cholesterol in our food, but there's actually high-density lipids, low-density lipids. High-density lipids are good. We want a little bit of cholesterol. There's some good cholesterol that we can have in our diet, and we're going to talk about that later on when we talk about the cell as well. But I want to give you a sense of this. Hormones can also have this structure. Things like estradiol, testosterone, other things like that. Again, some of those may come up, but for communication, but we're not going to be getting into deep detail in the endocrine system and other systems like an anatomy physiology class. But if you do come across anything in your independent studying and you have questions, send them my way. It doesn't have to be just about the talk. I'm happy to field those. So these are also a couple of other lipids, large molecules that come together. You can see here each of these are carbons, so these are even much bigger than sugars and lipids. Okay, so we had carbohydrates, lipids, and then now we have something called proteins, or little molecular machines of life. So this is when they really kind of start having form, meaning function, to an observable degree. And that form and function is another major theme that you want to think about as you move along. So we have this idea of structures building in complexity as little units combine to make larger units, but also the structure of individual things. It's not that they have a purpose, it's just that that structure imparts a function that life found a way to make use of. We're not quite at that whole level of evolution yet, because we're at the molecular level, but the principles of what works, sticks around, is something that does go throughout your AP Bio course. So before I move on, I know I want to address a couple of questions that we have here. For lipids, they don't really have monomer and polymer setups. You could think of the fatty acid as a monomer, and then 3 of those come together maybe, but really, since triglyceride is its own kind of thing as well, they're not forming chains so much. They're just molecules that are going to come together for a particular function in the cell. And then phospholipid, as an example, probably heard phospholipid membrane. That's where that's going to be a huge component. And then saturated and unsaturated, just to clarify that, yes, the presence of at least one double or triple bond where you don't have all the hydrogens you possibly could makes it unsaturated. You're not going to need to know a ton of organic chemistry. Organic meaning containing carbon, inorganic not containing carbon. Water, inorganic, carbohydrates, organic. You're going to only really need to think about this idea of how that structure gives the function. The idea of, oh, it's unsaturated, it has a bend, and what does that mean for where this is going to be useful in the cell? And then hydrophobic and hydrophilic, those are great terms. I actually get to those later on in my term card, but I'm going to mention them now. When something is polar, remember that's going to like polar solvents. So that those two things are going to go together. Hydrophilic would be the polar ends of things. So loving water, hydro water, philic is actually philia, love of. So that's love of water. And then hydrophobic, hydro, water, phobic, phobia, fear of. So or in this case, water hating, something like that. So I think that clarifies your questions. And my scroll wheel keeps getting away from me. But I want to talk about proteins now. So proteins, general structure. They are made up of a chain of amino acids. This is an amino group. Again, don't really need to know the names of these groups, but if you recognize this, a lot of the AMs go with nitrogen with hydrogen. So ammonium, NH4+, ammonia, NH3 without the, just on its own without the charge. Amide groups are NH2, and different iterations of how many hydrogens and how they bond and everything. Again, beyond the scope of the class, but amino acid, and then here's that carboxylic group again. It's missing its hydrogen because it already donated it, because that's what makes it an acid, amino acid. This group down here is the side chain, or R group, and that changes between every amino acid. How many amino acids are there? 20. Do you need to know all of them? Nope. So you don't need to worry about the various R groups for each one, or the different codes for, there's called single letter and triple letter codes and all that. We'll get to more of that in the molecular biology that we'll move on to, but I wanted to show you that general structure. And then how they link together. So you can see this box right here would be where one amino acid is, and then another one starts, and then another one starts. And I just know that by looking at the amino, and then, ooh, that C double bond O looks like this guy. So that's the other end, and then it comes here. That bond between that nitrogen and carbon is called a peptide bond, because proteins are also called many peptides, or many individual amino acids linked together. An amino acid is a peptide. What happened here in order to make this bond? Any wild guess in terms of the kind of synthesis that we've been mentioning? Dehydration synthesis. So what actually happened was, so actually I just want to, so this H is still missing, it's on the end over here. And for this bond to have happened, it couldn't have been there either. But you notice that the N only has one hydrogen on it, 2 are missing, and there's only one oxygen bonded to this carbon. So an oxygen is missing. This is another way in which dehydration synthesis happens, the formation of polypeptides. That's why it's so important. Proteins do all the heavy lifting in life, in our cells. And so, I'm trying to do this auto scroll thing here for the questions. So dehydration synthesis is once again rearing its head. That's why I mentioned these things. Now, something I didn't mention before that I can mention now, how would we split apart this polypeptide here into individual amino acids? We could reinsert the water molecule. The water molecule is capable of breaking that peptide bond. We call that hydrolysis or hydrolysis. Again, terms will be on the card at the end. But water breaking. Lyse is to break apart. And so hydrolysis is when water is the entity doing that. Okay, Beyond this idea of forming a chain, as you see here in the primary structure, we call that the primary structure. You may see that also as a 1 with a degree symbol, first degree. So the primary structure is just a sequence of amino acids. Another concept that is going to come up, has already been playing a role, and I'm going to draw attention to now, and I'm going to continue to draw attention to in future units, is how the local environment totally affects structures and functions of things. This idea of responding to the environment and processing what it means for the molecules, or the compounds, or the macromolecules, or the cell or the organism itself. That's that whole idea of information storage and transmission, that idea of energetics, inputs and outputs. So if I have a particular order of amino acids here, the order that they're in, the sequence that they're in, has an effect. And so that means these amino acids are impacting each other and going to end up changing the structure. They may want, like maybe amino acid 3 wants to be a little bit closer to amino acid 1 than it is, so it could make this what we call an alpha helix, where it forms kind of like the helix of DNA. It's not quite the same helix, but it forms a helix in order to bend around like that. Another structure that can form is called a beta pleated sheet, or you may just see it as a pleated sheet. But in this case, they kind of form these kinks in between and bend back and forth, and sometimes there are amino acids that are responsible for a turn. Again, we don't need to know that that's proline, but that's the amino acid that usually does that. These two structures are a result of the local interaction between the amino acids. Okay, let's bring it to the next level. What about if these interact with each other? That's the tertiary structure, the third degree. So this is when you start getting the actual structure of the protein. We would actually do something called X-ray crystallography and determine the atomic structure and space-filling model of the protein. And that's what's actually going on here. One further level is that when proteins come together, they can also interact with each other and make even larger macromolecules. That would be the quaternary structure. That's when different subunits come together, as in this example of hemoglobin. So these are heme units, and then this is the rest of the protein. This structure was determined many decades ago with technology nothing like what we have now, and it's pretty darn impressive how we figure out things of even this size, never mind at the lower size, and the fact that all the same properties that we observe in so many other places take place even at this level. Okay, one more thing I want to introduce that is then going to be the main star in our class next week are nucleic acids. There are two that you're going to need to know. There are different forms of each of them. We don't need to worry about that. Again, we're trying to take advantage of the fact that in class, you don't want to necessarily teach towards a test. It can be super dry and super boring, and you can miss out on all kinds of really interesting information. Something that you may not have known that then engages you further. But what's going on here is all we need to know is what we need to know in the test. Deoxyribose and ribose. So ribose would be the main sugar. You see here that's actually a 5-carbon sugar, not a 6-carbon sugar. Any ideas why we would call one deoxyribose and the other just ribose? So there we are missing the hydroxyl group, or the oxygen, essentially. So deoxy, D as in delete, as in remove, oxy as in oxygen. So that is now missing. And so this is called deoxyribose, and this is ribose. Want to bet what the D in DNA stands for? Deoxyribonucleic acid. So deoxyribo is the D, and that is this sugar here. And then RNA, ribonucleic acid, it's just this one. So those actually also lend themselves to very different structures. DNA tends to be double-stranded, RNA single-stranded. We'll talk about that more next week. This is an example of a nucleotide. So we have here the sugar that we were talking about. In this case, we don't have a hydroxide coming down. So it is, yep, I see that. We've got some good answers here from Alina about the 1 less hydroxyl group. So this right here is deoxyribose. So we know this is going to be a DNA nucleotide. This is that phosphate that we saw before. Now there's an H on there simply because I haven't donated it yet. But that's the same phosphate that we saw on phospholipids. And then this guy here is what we call a nitrogenous base. Lots of nitrogens, and then it's the base, as in ATGC, the bases in our nucleic acids. This one happens to be adenine, I believe. I don't need to know that off the top of my head because, one, I'm not taking a test, but you don't need to know it either because you don't need to know the structures of bases on your exam. So if you had a test, even maybe recently, because this may have come up in Unit 6, that had you write out the bases and know cytosine, guanine, adenine, thymine, you don't need to know that for the AP Bio exam. So what we have here is when we don't have the phosphate, so we have here the sugar, right? And then when we put on the base and we don't have the phosphate, that's a this guy right here, that's a nucleoside, it's S for sugar. And then the nucleotide is when we add that phosphate. This is also called monophosphate because there is one. But something like, so we would call this AMP, adenine monophosphate. You may have heard ATP, that's adenine triphosphate. There would be two more phosphates joined here with what we actually call oxygen linkages in between. But again, just to give you a sense of where you've actually seen this before and how it all comes together. So lots of terms. Again, I'm not going to go through all these, but the main things, this idea of monomers coming together to form polymers, this idea of carbohydrates, they happen to be linked by something called like glycosidic sugar. So they have glycosidic linkages between them. Lipids are fats. Some other carbohydrates that I hadn't mentioned, starch, pasta, that's basically a bunch of sugars. Glycogen, that's how we store our sugars. It comes in as glucose, and if we don't use it, we store it as glycogen. Cellulose, that's the stuff that we can't digest, actually, because they have different linkages than the sugars that we can. This is from plants. All of the outer layers and the rinds of plants. And plastids, less impact there, but that's how plants actually store their sugars as well. Hydrophobic and hydrophilic and hydrophobic, I told you I would mention those. Here they are. Amphipathic. So kind of like the word ambidextrous, two ways that you can, work with things, dexterity. So amphipathic is two, in this case, kind of pathologies. Path in that way means feeling. So an empathetic person feels what you're feeling. A sympathetic person understands what you're feeling. Doubtful there's anybody that's amphipathic, but amphipathic means that you both have a hydrophilic end and a hydrophobic end. This is actually how soap works. One in the soap is organic and hydrophobic. It takes all the grime, and then the other end is hydrophilic and it likes water. And so then when you rinse the soap off, it pulls the grime with it and it flows with the water because water has adhesive and cohesive properties. It's just another way in which we can actually see that this stuff has come up before. Okay. We also have amino acids here. I talked about the peptide bond and the structures. I couldn't possibly put in all that description here on the card, but you know to be aware of those peptide interactions. There was a question earlier about how is tertiary structure when alpha helix and beta pleated sheets come together. It's essentially just that next level. Like imagine a microscope throughout this course and you just keep dialing it back to zoom out more and more. We're zooming really far in and we can see the alpha helices and the beta pleated sheets, and then we zoom out a little bit more and now they're smaller players and we can see the larger protein. Those different structures are interacting with each other. So essentially, yes, it is when they come together, but it's not as though they're really independent. A given amino acid is going to have regions that have the alpha helices and that have beta pleated sheets. It's all dependent upon what the amino acid sequence is in that area. Those same different structures, you guessed it, then give that protein different functions. It could determine where it's going to be located, what it's going to interact with. We're going to talk later on about enzymes, which are these catalytic proteins that really get things going in terms of making reactions happen faster, but they're also super specific to what they actually can work with. And I also mentioned these couple of chemical reactions at the end as well. Any other questions at the moment? I think we're doing pretty well in pace. I just want to keep going and finish up with a question. So I'm going to give you a couple of minutes, but essentially identify, notice the bold there, the key metabolic processes for step 1, that'll be here, and key metabolic process for step 2, that'll be here, and briefly explain how each process promotes movement of carbon through the cycle. So this is the simplified carbon cycle, like they said. This is going to draw on a lot of other topics, but again, my expectation is that you've already seen a lot of those, but this is how a lot of the stuff we talked about today can come into play. This idea of things getting reused, broken down, built up, and put together. That's how I wanted you to think about this problem. So go ahead, and if you'd like, even if this is for the recording, you can pause it, take your time, and then play it. But I'll give you a minute, and then I'll show you the scoring guidelines and what to think as you go through this. Okay, so how would we go about this? Identify. So all I need to do for that is name something. That's all I need to do. I need to point out a key metabolic process for step 1 and the process for step 2. It actually says the, so it's going to be specific. If it said A, then there'd be more than one, and any one of the options would be fine so long as it's relevant, but you just need to name it to get that point. And then briefly explain how each process promotes movement of carbon through the cycle. Your explanation should focus on the role of energy. So they're telling you what they want you to focus on here, the role of energy in the movement of carbon. That's where you're actually going to be providing a little bit more of that shorthand, not shorthand, but short free response, prose, time test, different situation I know this year and all that. We're going to find out more information on Friday. But that's going to actually require putting together some sentences, which is why I also like to give you a little bit more information so that you feel comfortable and confident knowing the survey of the land and how to put that information together. And then for part B, just identify, again, just name, an organism that carries out both processes. So there are many different ones, and it's just any one of them works. And I really want you to pay attention to how I lay that out in terms of what you read, what to think, and then all the little details that I've been doing these exams for a long time. And honestly, they haven't even changed much when I took them. So we can take advantage of that. We can exploit that similarity, that standardization, and know what it is that they want us to do. Sure enough, here's my identification. In 1, CO2 going to organic molecules, just larger macromolecules. That would be photosynthesis, specifically the Calvin cycle. They're saying dash, so either one of those would be fine because they just wanted the process. The Calvin cycle is when CO2 is taken in triplet, 3 molecules at a time, and you run the Calvin cycle twice. Well, now you have 6 carbons. What can you make? Glucose. This is actually how plants make their sugar at the very end. It's the light-independent reactions. We used to call them the dark reactions in my day, but they actually don't actually occur in the dark. They just don't require the light the way the photosystems 1 and 2 do previously. And then in the other one where you break that down, cellular respiration. We specifically want the release of CO2. So glycolysis is not going to cut it. We don't lose CO2 there. But we do in the Krebs cycle, which is also known as the citric acid cycle. So at that stage inside the mitochondria, we let off CO2. Okay, and then what about our explanation? So remember what I said about inputs and outputs? I said it for a reason. You're talking about CO2 is fixed, which is basically means hydrogens are added to it. Again, you're not going to need to know the structures of these molecules, but if you see that term reduced or fixed, usually means hydrogens were added on, maybe oxygens removed. And then the role of energy in the movement of carbon. So we're using that light energy or ATP produced from the light reactions. I mentioned photosystem 1 and 2. That's what's going on there in order to get us to have the ability to run through the Calvin cycle. And then what do you get? I mean, organic molecules, glucose. Cellular respiration. In this case, they're hydrolyzed. Remember, hydrolysis, broken down. Water is coming in and breaking it apart. And then we're using that energy for any myriad because we're making energy. So we get ATP production or cells able to grow or anything like that. And then the output would be CO2. Okay. The identification. So I told you how they said and. There are multiple different options. Plants do both because of the photosynthesis and the Calvin cycle. Algae, anything really that is either photosynthetic or, in this case, a bacterium that fixes CO2, brings in CO2 for the second part to make those molecules. Lichen is a little bit different. The reason that they don't want you to say fungus here is because a fungus is a heterotroph. It eats on things outside. Hetero, other, troph is kind of like behavior, like trophic levels. You may have covered that in AP Bio. So not a fungus, but a lichen is actually a mix of photosynthetic and cellular respiration processes here. More on that later, but we don't really have to get into that either. So I hope that process helped. I hope that I sufficiently pointed out the important things to keep in mind, the way in which you want to think about the questions and work through them, how the content that we just had, while very foundational, is still relevant even on a question like this with the other bits of information we're going to cover coming up. And I hope that we see you next week for molecular biology and genetics, when we talk a lot more about those nucleic acids, DNA and RNA. So you're talking replication, transcription, translation, all that jazz. You have any questions? classes@prepmatters.com. This is also part of our PMTV series. So hashtag PMTV on social media, and we will see that stuff. And you can also follow us at PrepMatters. Thank you so much.